Hello everyone. So in this video tutorial we are going to describe about the reaction 5, 6, 7 and possibly 8, reaction number 8 of glycolysis. So in the reaction 4, that is the aldolase catalyzed reaction, this particular reaction catalyzes the conversion of the fructose 1,6 bisphosphate to two molecule. One is the dihydroxyacetone phosphate, another molecule is the glyceraldehyde 3-phosphate. So reaction number 5, which is the last reaction, or what do you say, the preparatory reaction, the last preparatory reaction of the glycolysis, and that is the conversion of this one molecule of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. So reaction number 5, which is a triose phosphate isomerase catalyzed reaction. Triose phosphate isomerase is the enzyme. You can see the name of the enzyme, triose phosphate isomerase. This enzyme actually catalyzes the conversion of this dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. So now after this reaction you have two molecule of glyceraldehyde 3-phosphate. Once the first glyceraldehyde 3-phosphate arises from the reaction 4 and the second glyceraldehyde 3-phosphate arises by this reaction. So because dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are basically ketose aldose isomer, and that is why we require an enzyme called isomerase, and it is a triose sugar because it contains 3 carbon atom, and not only the 3 carbon atom but it also contains a phosphate group, and that is the reason why it called triose phosphate isomerase enzyme. So this actually closes the preparatory stage, which is the reaction 1 to reaction 5 of glycolysis. Now we should move to the reaction 6 of glycolysis, that is the ATP generating stages of glycolysis. So reaction 6 to reaction 10 are basically known as the ATP generating stage of glycolysis. So this is the reaction number 6. So now from this reaction, all of the reaction will be multiplied by 2 because you have now 2 molecule of glyceraldehyde 3-phosphate. So the conversion of glyceraldehyde 3-phosphate to 1,3 bisphosphoglycerate is catalyzed by an enzyme called glyceraldehyde 3-phosphate dehydrogenase. So this is the reaction number 6 of glycolysis, or it is known as the first high energy intermediate formation. So in this reaction, what you can see, the substrate is the glyceraldehyde 3-phosphate, and we are using an oxidizing agent like NAD+. So after the reaction, NAD+ becomes the NADH. So because this reaction will occur in 2 units, so that means there must be 2 molecules of glyceraldehyde 3-phosphate, 2 molecules of NAD+, 2 molecules of inorganic phosphate to generate 2 molecules of 1,3-bisphosphoglyceride, 2 molecules of NADH, and 2 molecules of H+. So this phosphate will be taken up at the first carbon atom, that is the number 1 carbon atom. So this phosphate will replace this hydrogen attached to this carbon atom, resulting the formation of 1,3 bisphosphoglyceride. Why it is the bisphosphoglyceride, why not diphosphoglyceride? Because the position of the phosphate groups are located at the defined carbon. If the 2 phosphate groups are located on the same number of carbon atom, then it is called the diphosphate. Or maybe if the 3 phosphate group is attached to the same number of carbon atom, it is called the triphosphate, such as adenosine diphosphate or adenosine triphosphate, something like that. But if the 2 phosphate group or 2 same group attached to the different number of carbon atom, then it is called the bisphosphate, bisphosphate. So that is why the product is 1,3-bisphosphoglyceride. NAD+ acts as an oxidizing agent of this reaction. In any reaction, wherever you are using NAD+ or NADH or maybe FADH2, then the name of the enzyme will be the dehydrogenase. And because the substrate is glyceraldehyde 3-phosphate, so the enzyme is glyceraldehyde 3-phosphate dehydrogenase enzyme. So this is the particular reaction which involves direct hydrate transfer. That means the hydrogen is directly transferred to the NADH. If you level the hydrogen with the H3, that is tritium, in that case you can get leveled NADH molecule. So this is the particular reaction occurs in glycolysis, reaction number 6, which involves direct hydrate transfer, and the GPDH will be inactivated by the addition of iodoacetate molecule. So iodoacetate is ICH2CO-. So this is the iodoacetate molecule. If you add iodoacetate, that iodoacetate will rapidly bind to the GPDH active site. So there is some question that how can you establish the fact that GPDH active site contains cysteine sulfihydryl group? That means GPDH contains cysteine sulfihydryl group. So this is the structure of the enzyme, means all overall the glyceraldehyde 3-phosphate dehydrogenase, and the active site contains cysteine. We all know cysteine contains CH2SH in the form of their R group. So if we add iodoacetate, what will happen? This iodine and this hydrogen will be eluted in the form of HI, and as a result the formation of a complex. So if you digest the complex by some addition of some agent, in that case what will happen, the entire enzyme consists of many different amino acid will be eluted out as other amino acid, only the cysteine will be remain in the form of carboxymethyl cysteine. So if you get the carboxymethyl cysteine in the product, then other amino acid, this other amino acid means they are the simple amino acid like proline, maybe cysteine, maybe arginine, maybe glutamic acid, maybe lysine. These are the other amino acid that is not modified. Only the modified portion is the active site which contains cysteine molecule. So whenever you are adding iodoacetate into it, the product, that is the product which is a cysteine product, which is the cysteine is also associated with that acetate molecule. As a result, the product is the carboxymethyl cysteine. So this is the carboxyl group. So carboxy, CH2 means initially it was CH3, so carboxymethyl, and this is the cysteine part. So this is the carboxymethyl cysteine. So if you get carboxymethyl cysteine, then you can surely say the active site of the GPDH contains cysteine sulfate group, and it also explains that how iodoacetate will work, how iodoacetate actually act as an inhibitor of the GPDH reaction. So this is the particular reaction which involves direct hydride transfer. If you level the glycylate 3-phosphate by H3, that is tritium, at the number 1 carbon, in that case the product NADH will be radio labeled. So why this particular reaction is known as the first high energy intermediate formation? Because here the product contains 2 phosphate group, and now one of the phosphate group can be donated to the adenosine diphosphate to generate ATP molecule, and that is why it is called high energy intermediate, because it contains 2 phosphate group. One of the phosphate group can be donated to now to a different molecule like ATP. So this is the actually the which occurs in the reaction 7 of glycolysis. So this is the reaction number 7 of glycolysis, that is the first ATP generation reaction. So it starts with the 1,3-PG. It contains 2 different phosphate group at position 1 and position 3. So if you donate this phosphate group to the ATP molecule, resulting in the production of ATP. So the first reaction where ATP is generated by the glycolysis is the reaction 7 of glycolysis. Okay. So whenever we are transferring a phosphate group from ATP to a metabolite, or from a metabolite to ADP to generate ATP, once again, when you are transferring a phosphate group from ATP to a metabolite, or vice versa, that means to a metabolite such that here from a metabolite to ADP to generate ATP, then the compound will be the kinase enzyme. So kinase is a type of enzyme that transfer a phosphoryl group between ATP and a metabolite. So that is why, because the substrate contains phospho and it is the glycerate, that is the 1,3-bis phosphoglycerate, that is why the enzyme, name of the enzyme is the phosphoglycerate kinase, and it requires magnesium ion. Magnesium is always required wherever you are using ADP or ATP in this case. So there must be some sort of mechanism present. This is the mechanism. You can see 1,3-bis phosphoglycerate molecule, and this is the adenosine diphosphate. This is the alpha phosphate and this is the beta phosphate, and it is joined. This alpha and beta phosphate contains many negatively charged oxygen particle, and there must be some sort of repulsion between this oxygen. To stabilize it, you require a magnesium ion. So this oxygen is free, so they will nucleophilically attack this phosphate group, resulting in the cleavage of the phosphate oxygen bond. So this phosphate will be accepted by the adenosine diphosphate to form adenosine triphosphate. Now you can see the magnesium ion which is present on the alpha and beta carbon atom. Now it is shifted from alpha beta to beta gamma carbon atom, resulting in the production of the 3-phosphoglycerate. So this is the reaction mechanism. That is the reaction number 7. So now what you can see, a good example of what you can say, that is a coupled reaction. So reaction number 6, this is the reaction number 6, that is the conversion of glycerate 3-phosphate to 1,3-bis phosphoglycerate, and this is the reaction number 7 that just we have heard, that is the 1,3-bis phosphoglycerate to 3-phosphoglycerate. So in the first reaction, that is gap Pi plus NAD plus, resulting in the production of 1,3-PG and NADH. So this is the reaction 6. But the del G value of this reaction 6 is plus 6.7 kilojoule per mole. It means that whenever in a particular reaction the del G value is positive, this is not occur spontaneously. It is a non-spontaneous reaction, because for the spontaneity of every reaction, del G value is negative. That means reaction number 6 will not occur in a physiological concentration. Okay, but the reaction number 7, that is 1,3-BPG plus ADP is equal to 3PG plus ATP. This is the particular reaction. These two reactions are the reversible reaction basically. This reaction number 7 is highly negative. The del G value is minus 18.8. So what will happen? This reaction number 7 actually carry out the reaction number 6. So together the overall reaction, the del G value of this two overall reaction is minus 12.1. That is the plus 6.7 minus 18.8. So as a result, the final del G value, del G 0 dash value is minus 12.1 kilojoule per mole. So the left is gap plus Pi plus NAD plus. So you can say gap plus Pi plus NAD plus ADP. ADP is also there. 1,3-PG will not be present in the left side because 1,3-PG is a product in this case. So you can cancel out 1,3-PG from right and from left. So the product will be NADH, ATP and 3PG. So this is the product. So as a result, the overall reaction, del G value of the overall reaction is negative, and so this kind of reaction is known as the coupled reaction. Whenever the second reaction actually carrying out the first reaction, this is called the coupled reaction. Okay. So this is the reaction number 7. Now you should turn for the reaction number 8. So it is a very simple reaction basically. You have 3-phosphoglycerate. This is the reaction number 8. You have 3-phosphoglycerate molecule, and the 3-phosphoglycerate will be converted to 2-phosphoglycerate. So the phosphate group which is present at the position number 3 carbon atom will be shifted to position number 2. This is the part, position number 2. So whenever a group is moving from one part of a molecule to another part, it is known as the mutase enzyme. Okay, so in this case phosphate group is moving from this position number 3 to position number 2, or maybe if you transfer phosphate from position number 3 to position number 1, then the same enzyme will be present, that is the mutase. So mutase catalyzes the conversion of catalyzes the transfer of a group from one position to another within a molecule. So in this case you record phosphoglycerate mutase. So basically it looks a very simple reaction, but for this reaction you record a catalytic amount of 2,3 bisphosphoglycerate as a reaction primer. That means if you carry out this particular reaction, you record a 2,3 bisphosphoglycerate in a very small concentration. So the question is, how 2,3 bisphosphoglycerate arises? So what we see here, that is just the transfer of phosphate group from position 3 to position 2, but that is not actually happening. This is the details mechanism actually. So this is the enzyme which is the phosphoglycerate mutase enzyme, and it contains histidine at the active site, and it is not only the histidine but also a phosphate group is associated with the histidine molecule, and as a result this enzyme is called phosphoenzyme. That means the enzyme already contains a phosphate group. Now the substrate is coming, that is the 3-phosphoglycerate. So this is the 3-phosphoglycerate. Initially you have an idea that the phosphate group is actually moving from position 3 to position 2, but that is not actually happening. Actually this 3-phosphoglycerate is taking this phosphate group from the enzyme active site, resulting the formation of 2,3 bisphosphoglycerate initially, and after formation of 2,3 bisphosphoglycerate it will donate the phosphate group from position 3 to histidine back again, so that now you have this 2PG complex. The 2PG will be released from the enzyme active site. So initially what we are thinking, that the phosphate group may be shifted from position number 3 to position number 2, but this is not actually the case. The phosphate group is actually present at position number 3 initially, and they will take one phosphate group present already in the enzyme active site to generate the formation of 2,3 bisphosphoglycerate, and now whenever they have the 2,3 bisphosphoglycerate, they will donate the phosphate group from position 3 to histidine back again, so that now you have 2PG phosphoenzyme complex, and from there 2PG will be eluted out, resulting the formation of the phosphoenzyme back again. So if this 2,3 bisphosphoglycerate gets released from this active site, in that case it will be the inactivated enzyme because this lacks the phosphate group. So from this you can say that 2,3 bisphosphoglycerate may be generated and may act as a reaction primer to carry out the reaction. Now one of the important thing to note that what you can understand, that 2,3 bisphosphoglycerate is getting produced by the conversion of this particular reaction as an intermediate, that is from the 3-phosphoglycerate to 2-phosphoglycerate conversion by the formation of an intermediary compound, 2,3 bisphosphoglycerate is generated. But in erythrocytic cell, that means in RBC cell, the concentration of 2,3 ppg is much higher, that is nearly 5 millimolar, then trace amount is required for use as a primer for the phosphoglyceride mutase reaction. That means in RBC cell, the concentration of 2,3 bisphosphoglyceride is much higher than any other cell. So why in case of RBC cell the 2,3 bisphosphoglyceride is actually higher? Because in RBC cell they have an alternating pathway for the generation of 2,3 bisphosphoglycerides. So normally what you can see, normally from the conversion of 3Pg to 2Pg conversion, 3Pg to 2Pg conversion, there must be some sort of intermediary compound, that is the 2,3 bisphosphoglyceride. But in this particular reaction, in RBC cell, two different enzymes are also present, that is absolutely present in RBC cell but not in other cell. So RBC cell contain two different enzymes. One is the bisphosphoglyceride mutase, which catalyzes the conversion of 1, bisphosphoglyceride to 2, bisphosphoglyceride first, and then there is a phosphatase, that is 2, bisphosphoglyceride phosphatase, which will now clip the phosphate group at position 2 to generate 3Pg. So that means what we can say, RBC must have a detour from the normal pathway. So there is basically two ways present in RBC cell by which they can generate 2, 3 bisphosphoglyceride. The first is, like other cell, they can generate 2, 3 bisphosphoglyceride as an intermediate during the conversion of 3Pg to 2Pg. From the 3Pg to 2Pg conversion, they can generate 2, 3 bisphosphoglyceride as an intermediary compound, and there is another pathway, that is the conversion of 1,3-BPG to 1,3-BPG to 3Pg conversion. They can synthesize 2, 3 bisphosphoglyceride because they have two different enzymes. So this is the way by which RBC cell must produces much higher concentration of 2, 3 BPG than other cell. So why they are producing much higher concentration of 2, 3 BPG? Now we have understood that how they can produce this 2, 3 BPG in the cell in higher concentration. In the cell means in the RBC cell, definitely. In the RBC cell, the concentration of 2, 3 BPG is much higher, that is nearly 5 millimolar. So how they can produce this? They have the two pathways. One is the conversion of during 1,3-BPG to 3Pg, they can generate the 2,3 BPG, and the second conversion is the 3Pg to 2Pg conversion, they can also generate 2, 3 BPG. But now the question is why they are producing? Because hemoglobin, which normally transport oxygen to all parts of the respiring tissue, can also bind with the 2, 3 BPG significantly. So the idea is 2, 3 BPG specifically binds to the deoxyhemoglobin and thereby alters the oxygen affinity to hemoglobin. That means if you have hemoglobin, normally what will happen, the deoxyhemoglobin must bind to oxygen to form oxyhemoglobin so that the oxygen can be carried to all parts of the respiring tissue. But if the deoxyhemoglobin sees 2, 3 BPG is also present in the system, in that case 2, 3 BPG can competitively bind to the hemoglobin so that oxygen is not able to bind. Okay, so that means you have hemoglobin and you have two components, one is the oxygen, another is the 2, 3 BPG. So it is a concentration dependent process. If the concentration of 2, 3 BPG rises, then 2, 3 BPG rapidly bind to the hemoglobin molecule, and as a result hemoglobin will not able to carry out oxygen. And if you have a significant concentration of oxygen, in that case hemoglobin can carry oxygen, not the 2, 3 BPG. So in case of hexokinase deficient individual, a person that is a deficient of hexokinase, that means hexokinase which catalyzes the first reaction of glycolysis, and if the person is deficient of the hexokinase, it means the rate of synthesis of 2, 3 BPG will be significantly lower in those individuals, because the first reaction is not carried upon in a significant rate, so the second reaction will not be carried on, the third reaction will not be carried on, and as a result this particular two reaction will not be carried on in significant concentration, and as a result in those individuals the concentration of 2, 3 BPG is actually significantly less amount. Because it is in less concentration, what will happen, the erythrocytic cell now can actually carry out, the hemoglobin actually now can carry out a significant concentration of oxygen in the form of oxyhemoglobin. But if a person is deficient of pyruvate kinase, so this is the point, pyruvate kinase deficiency. So pyruvate kinase deficiency. So if a person is if a deficiency of pyruvate kinase, in that case all of the pyruvate kinase means it is the last reaction of the glycolysis. Reaction 10 of glycolysis will be carried out by the pyruvate kinase reaction. So if the last reaction is not carried on significantly, in that case there will be a significant concentration of accumulated 2, 3 BPG by these two pathways. because the last reaction will not carried on. That means there is the accumulation of reaction number 9, reaction number 8, reaction number 7, and also the intermediary component. So this is the two intermediary component. One is the 2, 3 ppg, another is the 2, 3 ppg by this conversion, 3 pg to 2 pg conversion. So if the pyruvate kinase is deficient, if an individual has a deficiency of pyruvate kinase, that means the last reaction is not carrying out in significant amount. As a result, there will be some accumulation of the products in the previous reaction, that is 2 pg will be accumulated, 2, 3 ppg will be accumulated, 3 pg will be accumulated. So in that case, those cells have a higher concentration of 2, 3 ppg, and as a result, that hemoglobin will not be able to carry out oxygen significantly. Okay, so this is the actual mechanism that why we require 2, 3 ppg, or why 2, 3 ppg actually inhibits the oxygen transport. What will happen in hexokinase deficient individual, what will happen in pyruvate kinase deficient individual. So for hexokinase deficient individual, this is the graph, the green color graph. For pyruvate kinase deficient individual, this should be the graph, and this is the normal individual. So in case of pyruvate kinase deficient individual, they will not carry oxygen significantly because the hemoglobin rapidly binds to the 2, 3 ppg. But in hexokinase deficient individual, what will happen? The concentration of 2, 3 ppg is very low. In that case, there will be high amount of oxygen transport by the hemoglobin molecule. So from this, we can end the reaction 8 of glycolysis. In the next video tutorial, we are going to describe about the reaction number 9 and the reaction number 10 of glycolysis. Thank you.